Hello, hello, hello. It is time for AP Bio Review with me, Mr. W. Time for AP Bio Review. That's what we are going to do. I am so glad to be here with you, broadcasting from the world headquarters of learn-biology.com and the Science Music Videos channel here in Berkeley, California. Berkeley High School was covered in the New York Times today because it's an incredibly wonderful, dynamic place. I had an amazing, amazing career there, and I am so happy to be there now. Anyhow, I am really, really psyched for tonight's show and for the shows that we're going to be doing over the next week because it is time. It is time to knuckle down. It is time to get serious. It is time to show who you are, because the AP Bio exam is just 1 week away. So I'm really hoping that we're going to have a fabulous show tonight. I'm hoping that we get lots of people here, but whoever's here, we're going to have a fantastic time reviewing, learning my favorite subject, biology. And I am so happy that the people who are here. It's fantastic. What would be wonderful would be just for us to get to know one another a little bit and for me to have a sense of who I'm teaching, for people to just use the comment on YouTube and just tell me who they are and where they're from. And that gives me a little bit of sense of who the people are who have come to do this incredible thing, which is to review for AP Biology. So again, I am so happy to have you here with me. And I'm actually so honored that you decided to take your time tonight to actually learn with me. So that's fabulous. So let's do this. I'll tell you about my plan for tonight. First thing tonight is that I wanted to inaugurate our community. And I thought that it would be good for us to give us a name. We are Biomanians. We are Biomanians. That's named after my app, but also we are maniacs for biology. Maniacs. I don't know if that's exactly the right word. I got to think that one through. But we are just filled with love for biology. And over the past weeks, we've already become a community. I know many of you who have chatted in. So hello, Ella, Sophie, Lily. It's great to have you here with me tonight. And here's the plan. And I hope this is a plan that you feel good about. I recently put out a 2-week plan for review. I think that everyone who I'm seeing so far already has that plan, but I'm noticing that there are more people here who haven't chatted in. So it's possible that it might still be useful to briefly go over that plan and talk about a way to wind your way through the next, week and 1 day, because that's really all you have to study. The test is on the 16th, so you have until the 15th to study. And I do want to say, like, wherever you are in your study process, 8 days of study with a weekend, you can learn an enormous amount, and you can do an enormous amount to prepare yourself, even if you're coming from a classroom where you didn't get good preparation. And from the people who I know who are watching on the stream, I know many of you have had exceptional preparation, which means that what you're doing is you're preparing yourself to make a difference between a 3, which is an attainable score, to maybe get a 4 or a 5 on the AP Bio exam. And of course, that's the thing that I always strove for my students. That was those are the scores of distinction that I always felt really good about. I am going to do a little change here, and that change is going to be like this. Fantastic. Okay, so I've already talked about the fact that we are Biomanians. I'm going to talk about our 2-week plan for review. If you haven't downloaded your plan yet, you can get one at apbiosuccess.com/plan. That's pretty easy to remember. apbiosuccess.com/plan. I'm going to revise that plan, and I'll send you an email once I have the revised plan, because you'll give me your email in order to get the plan. if I don't have it already. So I'm going to give you a 1-week plan, but you can sign up and get this 2-week plan, and I'll give you a preview of what that's like. And then what I thought we would do is we would start a process of basically reviewing the course and then responding to questions. And then what I didn't add here, it should have been on the list, is we'll spend some time doing some multiple choice practice. I want to do an FRQ session, and I've been doing that in the Zoom reviews that I have because it's just so much easier to do the kind of back and forth discussion that you need to do. But what I'm thinking about doing for one of the sessions coming up is just doing a walkthrough about like how you think, how you should be thinking as you respond to these FRQs. And there are all these published FRQs, which is a wonderful thing on the part of the College Board, but those are still hard. They are hard questions. I struggle with them myself. So I thought that might be useful. So that is the plan for tonight. And before I talk about the checklist, I wanted to just share something with you, and I've got to grab a link. I'm going to do that here. I'm going to go back to where we were, which I think is, oh boy, where were we? Not there. Here. Fantastic. Okay. So I'm going to go like that. Okay. Fantastic. So this is the plan. And I'm thinking, yeah, everybody can see that plan. That's good. And this is a Google Doc. And the first part is a little bit of introduction, but the second part is this. What you should be doing over the next 8 days is you should be finding the gaps in your knowledge and then filling the gaps. Find the gaps, fill the gaps, and also practice the actual skill. So what I've done in this 2-week plan, which I will develop into a 1-week plan, is I tell you how to first find the gaps. And you can do that through a checklist that I'm going to share with you in a second. And you can do it with quizzes that are available to subscribers at learn-biology.com. And again, to subscribe, what is it? It's a $24.95 monthly subscription, which might add up to a lot if you were going to be with me for the whole year, but you're not going to be with me for the whole year. You're going to be with me for a week, which means $24.95 is your only cost, essentially. So you would subscribe to learn-biology.com for $24.95. You would take 2 50-question tests. You would use the results from that test and the checklist to assess right now where your gaps are. What are the topics where you're strong? What are the topics where you're weak? And then what you would do is you would use flashcards. That was originally when there were 11 days to go, but I'm going to have to revise this. And then you're going to spend a lot of time writing FRQs and doing self-scoring, because that is in some ways the ultimate task that's associated with AP Biology. The multiple choice part is tough, but I think that with enough practice, you can get yourself ready for the multiple choice test. And I guess the same is true of the FRQ portion. Practice and you'll get there. And you have to practice the task. And somebody asked me last night, like, what can I do to get ready for the FRQ portion? Respond to FRQs. The College Board has dozens of them on their website. There are links to them on this plan. I also have over 100 FRQs. They're super short and they have great data sets. So again, that's something that you get access to when you subscribe to learn-biology.com. So this is the plan day by day. Every day you're writing, every day you are finding the gaps, every day you're reflecting on where you are, and then you're doing that in a recursive way. And that's how you get yourself up to the level where you can get a 4 or a 5. Somebody recently asked me why I don't just say a 5. And I think that's because a 5 is hard, really hard. And I've had some students who were extraordinary who got 4s, and I respect them and love them. And I will respect you and love you if you get a 4, because, things can go wrong, or there can just be like the one gap that you didn't fill or something like that. So 4 or 5, those are the grades of distinction that I go for. And those are actually the grades of distinction that you should go for. If you get yourself a 4 on the test, I want to see you go like this. I want to see you pat yourself on the back, and I hope your parents are going to be wonderful to you. If you get that 4, 4 is a wonderful score. So 4 or 5. And actually, if it were earlier in the year, I would offer that with a guarantee. a money-back guarantee that if you didn't get a 4 or 5 on the test, I'd give you your money back, but that would be based on you completing the curriculum, and you don't have time to do that. Biology is big. So that's the plan. Sign up for the plan on, bam, can go like that. Sign up for the plan on apbiosuccess.com/plan, and that's it. Now, for tonight, what we're going to do is something that you might have experienced a little bit already, because I recently put out a video that was called an AP Bio Speed Review. I'm not the only person on YouTube who has one, but mine goes with this checklist. And you can get this checklist by going to apbiosuccess.com/checklist. And since completing a checklist is the first part of a plan, I would suggest that you download this checklist right now if you don't have it. The checklist looks like this. Here, I'm going to grab the checklist, going to go like this, grabbing that, and I'm going back to our thing over here. That's where I am. Okay, here's a checklist. And the checklist basically walks you through the entire course. I mean, what more can I say? It's a checklist. I'm going to make that a little bit bigger. It's checklist. And so you go through the checklist and you check off what you know and what you don't know. And there's actually a method called a stoplight method, which is something that goes back years and years in education, where you mark things that you know well in green. Hey, relax, you got it. Things that you kind of know, you mark in yellow, and things that you need more help on, you mark in red. Then you go and you study the red. Okay, so this is the checklist. I have an introduction to it where I explain the stoplight method. And again, if you're not on it, go there now. I'm going to pop the URL in the chat. Here we go. Bam, bam. I'm not going to pop it in the chat. I'm just going to show it on the screen. But there it is, apbiosuccess.com/checklist. And that'll take you to a sign-up form where you'll give me your email address. And that way I'll be able to communicate with you about important things. And I'll be able to do what I'm doing now, which is encourage you to be a biomanian. Because what do biomanians do? Biomanians do what they need to do to get ready for the AP Bio exam. Biomanians are willing to pay $24.95 to get a subscription to learn-biology.com and the Biomania AP Bio app. Okay, let's do this. Your job is to pay close attention and also to interrupt. Yeah, that's right. Use the comments whenever there's a point that I hit on that you're not familiar with. All right, that's the whole point of this. And I'll go through a bunch of slides. I'll stop. I'll check for understanding. When it's possible, I'll do some interactive things. And then again, we'll leave about 25 minutes or so at the end to just go on to learn-biology.com, do some multiple choice practice so we can really get a sense of whether we know this stuff or whether we don't. Okay? So any comments, questions about anything? Like, was anybody hoping for something to happen that isn't happening yet in this show tonight? Because if it is, you know, it's just us and we can change direction. So anybody need anything in particular? And again, one thing I want to emphasize is that if you sign up for learn-biology.com, you get to do this with me on Zoom. And then when I ask a question like, do you need anything? You can just immediately respond and there's no 20-second latency. I'm very grateful to YouTube for providing this because, heck, it's great to be able to work with people around the world. But, you know, here we are. So I'm wondering what would be the best place for me to be. And how about, can I be there? That's pretty good. Okay. So we're going to start with a review of the solvent of life, which is water. And what do you need to know about water? Water is a polar molecule. Because it's a polar molecule, it forms, let me change my cursor here. Yeah, it forms hydrogen bonds. Hydrogen bonds are between the slightly positive side of one water molecule and the slightly negative side of another water molecule. Hydrogen bonds, I want you to remember, are weak bonds, much weaker than covalent bonds or ionic bonds. They're intermolecular bonds. They're between molecules, not within molecules. So I have this bad habit, which is when I do these slides, I get very, very excited. And I go ahead and I start telling you what is in the bullet points before I actually have a chance to deliver the bullet points. But I'm going to put myself over here. There we go. So water's a polar molecule. Already said that. Hydrogen bonds are intermolecular bonds, much weaker than covalent or ionic bonds. Because of water's polarity and hydrogen bonding, water has all of these incredible properties. One is cohesion. Water molecules cohere with one another. They adhere to other polar things. So here is a diagram that's meant to represent water moving up the very thin conductive tubes that make up the stem or part of the stem of plants. Water is partly able to do that because these walls of those tubes are made of cellulose. Cellulose is polar, so water can form bonds with those polar bonds because water's polar and it can form hydrogen bonds with anything else that's polar. There's also phenomena like surface tension. And there's others that are sort of more chemical and physical related, but for biology, if you know about cohesion, adhesion, and surface tension, you're pretty much set. So that's water. Now let's move on and talk about the elements of life, monomers and polymers, some very basic biological chemistry. The molecules of life are all built from monomers that combine into polymers. Legos are the monomers. The things that you build out of Lego are the polymers. And Aaron, I like that question. Thanks a lot. Very good. Thanks for getting this interactive. So when answering a question on the exam that asks how capillary action works, we can just say that cohesion and adhesion allow it. I would just throw in also the fact that because water is polar, right? So I would identify that water is polar. It will form bonds with the polar walls of they're called xylem, the tubes that make up plants. And as water does that, all the water molecules will grab onto one another. So it's those two forces. But like if this were an FRQ, I am positive that in the FRQ scoring guide, it would say something like student mentions polarity. Student mentions hydrogen bonding. So don't just say cohesion and adhesion. Say that because water is polar, it forms hydrogen bonds. That enables water molecules to have cohesion with one another and adhesion with other substances. And then I'd go from there. So don't hold back on your knowledge when you're answering FRQs. And, you know, of course, look for the right answer when you're answering the multiple choice questions. So Aaron, welcome tonight. I don't know if I've seen you before, and it's great to have you here. And thank you very much for that question. Any other questions about water hydrogen bonding before we move forward? I want you to notice that I'm drinking my tea from a Science Music Videos mug, but I don't want you to buy my Science Music Videos mug nearly as much as I want you to subscribe to learn-biology.com. Okay, let's go on. Molecules of life are built from monomers that combine. Wait, so how do we mention polarity? Vera, haven't met you before either. Fantastic to have you here. So how do you mention polarity? How do you mention polarity? Well, let's go back to Aaron's question. If Aaron was asking, you know, how do we explain capillary action? I don't really have a thin tube or anything to display anything, but like capillary action, capillary action is if you take a little thin tube, they're actually called capillary tubes, and you put it into water, the water will crawl up the inside of the tube. And you would do it in the way that I just modeled. So if the question were, a student notices that when she places a capillary tube, you know, it's like a very thin straw in a body of water, the water is able to crawl up the inside of the tube. Explain. Here'd be my explanation. Capillary action happens because water is polar. Because water is polar and the inside of the tube is polar, and it must be for capillary action to work, then water molecules form hydrogen bonds with the molecules that make up the inside of the tube. At the same time, as they're forming hydrogen bonds with the inside of the tube, which is bringing them up the tube, they're also forming hydrogen bonds with the other molecules other water molecules in that body of water. So those two forces, adhesion with the side of the tube, cohesion with water molecules and other molecules, water can make it up a short distance. How far? A centimeter, 2 centimeters. And Milkshaker, all these people who I haven't met before. This is so fantastic. What does plant grass cell wall look like? Is it a glucose polymer? Yeah, so Milkshaker, a glucose polymer means it's cellulose. It's, cell walls are made of cellulose, and cellulose is a glucose polymer. And heck, that's what we're looking at. And in fact, this is so cool. We're looking at glucose right here. Like, this is a carbohydrate monomer. So these are carbohydrate polymers, and you see these hydroxyl groups. Not really something that you have to know for AP Bio, but you kind of do, because this is what makes glucose or any monosaccharide polar. This is why sugar dissolves in water, because it has these hydroxyl groups that enable it to dissolve in water. So Milkshaker, a plant grass cell wall looks like this, but this is simplified. So it would still have these hydroxyl groups, and water could form bonds with these hydroxyl groups. So, wow, this is so super fun. We're going a little bit more slowly than I thought, but that's good. It's good because you're asking great questions. Okay, so I'm going to go on. Milkshaker, I hope that helped. And Milkshaker, maybe like your name is Mike or something like that, but I can call you Milkshaker. That's fine. Now, how do you put these monomers together? Monomers get put together through a process called dehydration synthesis. And in dehydration synthesis, what happens is enzymes, which aren't shown here, enzymes will grab two monomers, they'll fit into the enzyme's active site, and they will catalyze a reaction between those two monomers. And that reaction will involve forming a bond between this oxygen over here and this like stub of a molecule over here, and they'll pull out this OH and this H. OH plus H, that's H2O. So that's why a water molecule comes out. It's a dehydration synthesis, all right? So that's how monomers get put together, by a dehydration synthesis, and that removes a water molecule from the two monomers. The opposite process is hydrolysis. Hydrolysis is where an enzyme will come and break apart polymers into monomers. And as they do that, they'll jam a water molecule in to reform this OH over here and this H over here. Now, dehydration synthesis, what the heck is that? Everything you've learned about. So like, for example, when ribosomes are stringing together amino acids to make proteins, that's a dehydration synthesis. I'm almost positive that when DNA polymerase is stringing together DNA nucleotides to make new DNA during DNA replication, that's dehydration synthesis. When plants are taking the products of the Calvin cycle and they're putting it together to make monosaccharides, and then they're making those monosaccharides into polysaccharides like starch and cellulose, that's dehydration synthesis. So it's everything. It's everything that we've talked about. And hydrolysis is, when you're breaking down, let's see, in the cell communication unit, you've learned about how glycogen gets broken down to release glucose in response to a signal like epinephrine. Well, that is hydrolysis. So these reactions are everywhere. All right, so dehydration synthesis, that's how we build things in biology, and hydrolysis, that's how we pull things apart. So that's the basic chemistry. Now let's talk about some of the molecules that make up life. Protein building is through peptide bonds, right? Sophie, that's exactly right. But those peptide bonds are just a special name that we use for the bond that forms between two amino acids. So the bond between two amino acids is, is it exactly like this? I think it's actually going to come up in just a little bit. But when ribosomes build proteins and they're stringing amino acids together, I believe that is also a dehydration synthesis reaction. And the special bond that forms is a peptide bond, and that is about the only bond name that you need to know. Like, for example, the bonds that hold together a triglyceride, a fat, they have names, but you don't need to know them because that's too specialized for AP biology. But the one specific bond that you do need to know is the bond that holds amino acids together, and that's a peptide bond. And that's useful because, like, for example, protein, before it folds up into its specific shape, it's a polypeptide. Okay, so thank you, Sophie, for that question. All right, carbohydrates and lipids. The reason why I made that funny expression just now was you don't need to know that much about carbohydrates and lipids, so let's go pretty fast. So carbohydrates, there are the monomers, monosaccharides, simple sugars, disaccharides, two linked sugars, polysaccharides like starch and cellulose. Where do you see disaccharides? Well, if you've learned about things like lactose intolerance, an important evolutionary phenomenon among humans. Many humans can't digest lactose. Lactose is the disaccharide that's in milk. Many humans can't digest it beyond infancy. And that's because our enzymes for digesting lactose turn off after infancy. That's true of all mammals, but there are a couple of mutant humans. I'm kind of being tongue-in-cheek when I'm saying that, but there are a couple of populations of humans that maintained the ability to produce the enzyme that breaks lactose into monosaccharides. And they created all of these, sheep and goat and cow oriented cultures that emerged through herders in the, I don't know, a long time ago, 5, 6, 7,000 years ago, like, as you guys would say, way back in the day. All right, polysaccharides, starch, and cellulose. And one thing to know is that, like, for example, we have enzymes that can hydrolyze. You hear how I said that? Hydrolyze these bonds that hold together the glucose monomers in starch. And that's why all of our staple foods in our civilization are starches, like rice filled with starch, wheat filled with starch, corn filled with starch, right? So all of those starchy foods, that's how we get the calories that civilization runs on. But cellulose is another carbohydrate polysaccharide, but we don't have the bonds that can break the way that those sugar molecules are linked together. So that's why we can't eat paper and we can't eat wood. And when we eat things like celery, which has a lot of cellulose, we barely get any calories. We can't get much food energy from it. But some of our animal friends can. So, like, for example, cows can, they have a symbiotic relationship with bacteria that break apart cellulose. And that's why cows stand out in the field all day chewing their cud. I'm really getting off track. I'm really sorry. I got to stay focused. Monosaccharides for energy sources and building blocks. Disaccharides are for energy transfer, foods like sucrose and lactose, or I should have said sugars like sucrose and lactose. And polysaccharides can be used for energy storage. Starch is a plant energy storage molecule. A potato full of starch. Glycogen is stored by you in your liver and your muscle tissue when you take in too much sugar. That's what insulin does. Insulin gets your liver cells to take up glucose and convert it into glycogen. And then there are structural polysaccharides like cellulose, which makes up plant cell walls. And those plant cell walls, as I'm trying to remember who asked me about that, as Milkshaker asked before. Those are composed of cellulose. All right, lipids. We're going to cruise through these. Lipids, the thing about lipids is that they're non-polar, either completely or partially. They're unlike the other biomolecules because they're not really composed of repeating monomers. Whatever you mean by that. So look at this. Here's a carbohydrate, a polysaccharide, monomer, 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 all the way down. Lipids ain't like that. Lipids are just like, a couple of pieces put together. So here's a fat. That's for energy storage. Here's wax used for waterproofing. And I'm not going to teach how to interpret this other than say, like, every angle vertex is a carbon, and the carbon is assumed to have plenty of hydrogens attached to it. Phospholipids make up membranes. We're going to talk a lot more about those. And then molecules like steroids are used for signaling. Phospholipids, let's talk about those a little bit more. Phospholipids, hydrodynamics. Milkshaker just asked a cool question. Does saturated versus unsaturated affect the hydrodynamics? You might be using hydrodynamics in a way that I'm not used to it, but I'll tell you what saturated versus unsaturated does. So here's a phospholipid. It's got a hydrophobic nonpolar tail. It's got a hydrophilic polar head. That's partly due to this phosphate group right over here. Now, it's hung together by this glycerol molecule, but in relationship to your question, Milkshaker, this is a saturated fatty acid. This is an unsaturated fatty acid. Molecules with saturated fatty acids tend to be more solid. So like, for example, butter, saturated fatty acids. And butter is a solid at room temperature. Oils like olive oil and canola oil and all those other oils, they have more unsaturated fatty acids, and they tend to be liquid at room temperature. So that's what it affects. I don't know what you mean by hydrodynamics, but it does affect the solidity of the molecule. Basically, more saturated, more solid. More unsaturated, more liquid. I could give you many examples of that, but I'm trying to be super focused. When you mix phospholipids with water, they form a phospholipid bilayer, this structure. The heads interact with water. The tails form this water-free zone. Water-free zone. And then there's water inside. This, of course, is the framework of the cell membrane. So when we talk about membranes, we'll talk about this. All right, proteins. Any other questions so far? Though what I do want to emphasize is that, like, carbs and lipids, not a big part of the exam. You definitely need to know about proteins. Okay, so good, Milkshaker, for clarifying that question. These are both hydrophobic. Whether you're saturated or unsaturated, you're still hydrophobic. But so what the saturated and unsaturated thing is, it's a different access. So like, for example, oil, unsaturated, right? Doesn't mix with water. Fat, saturated, doesn't mix with water. They're both hydrophobic, but fats, which are saturated, tend to be solid at room temperature. You throw butter into the frying pan, it melts. Oils at room temperature tend to be liquid. So it depends, it determines like what state of matter it is. They're both equally hydrophobic. And Milkshaker, I'm loving these questions. Thank you. Okay, proteins. Proteins are the action molecules of life. They're the molecules that get things done. So proteins are responsible for muscle. But what's muscle about? It's not just about looking good. It's about motion. It's about contraction. Muscle tissue contracts. It's protein. Cilia and flagella, which are tiny little protein extensions of cells. They enable cells to move. Enzymes they break things apart. Hydrolysis, dehydration, synthesis, that's all enzymes. Structures, my fingernails. In fact, everything you're seeing on screen with me, it's all protein. My skin is protein, my eyes are protein, cartilage, is that protein? Okay, I'm not really an anatomy person, but skin is protein, fingernails are protein, eyes are protein. Protein makes the structures in an animal. In a plant, it actually is more carbohydrate. But transport, hemoglobin is a molecule that carries oxygen. Energy storage, the white of an egg, pure protein. And also transmitting signals. And I have a picture of insulin, a hormone that you should know and that we'll review when we do AP Bio Unit 4. When we do AP Bio Unit 4. All right. So the monomers of proteins are amino acids, and they have a central carbon. They have an amine over here. They have a carboxyl group. The amine is basic. The carboxyl group is acidic. Hence, they're amino acids. The name tells you what they are. And then they have a hydrogen atom. So those are all attached to the central carbon. And then they've got a variable group over here. And there are 20 varieties of that variable group. You don't have to memorize a thing related to that. But you do have to know that it's important whether it is polar, nonpolar, acidic, or basic. And you can tell that based on the functional group. And if you want to review functional groups, I'm not going to do that now, but go to learn-biology.com, sign up for a subscription, and I've got a fantastic tutorial about functional groups on learn-biology.com. So that's how I would advise you to do that. And I wouldn't spend a lot of time on it, and it's not, you don't have to memorize the names, but you need to know that, like, if this functional group is attached to protein, if it were a part of the side chain, then it would be acidic because this is a carboxylic acid. There are 4 levels of protein structure. The first is the genetically determined sequence of amino acids. So here we've got 3 amino acids linked together. I'm just checking to make sure that's the right number. Yeah, it is. And now I'm confirming what I thought before, that I'm pretty positive it is a dehydration synthesis when ribosomes put together amino acids to create, as Sophie said, peptide bonds. So this is the genetically determined structure. The secondary structure is interactions involving the polypeptide backbone. So this is the polypeptide backbone. Carbon-carbon nitrogen, carbon-carbon nitrogen, with hydrogens and carboxyl groups, I misspoke there, carbonyl groups and amino groups attached. Those carbonyl groups and amino groups, those can interact. And I think for AP biology, it's probably sufficient for you to know that they can interact in 2 ways. They can cause the whole polypeptide structure to coil up into what's called an alpha helix. Or if the polypeptide backbone bends back on itself, so that, like, here's one part of the here's one part of the polypeptide backbone, here's another part, and then here's another part, right? I'm going to go like this to demonstrate this. Okay, like this. So if the 2 parts of the polypeptide get close to one another, they can form hydrogen bonds between one another, and that can create a structure called a beta pleated sheet. That can happen if they're parallel to one another, or like this, anti-parallel to one another. I don't know how important it is for you to know the actual details of that, but what you do need to know is that, like, this kind of corkscrew structure, that is an alpha helix, and this is a pleated sheet, a beta pleated sheet over here. Now, interactions between R groups cause the molecule to form these hairpin turns and bends. And that actually can create the kind of interaction between parts of the polypeptide backbone that I was just talking about. So that is what we have over here, as Milkshaker just pointed out, a globular protein. And then when multiple polypeptides come together, then we have a quaternary structure. And I think you got to know all 4 of those. And I've got a couple of videos about that. But again, best way to know about that is $24.95. Then go to learn-biology.com, do the tutorial about this if you didn't get it in your class, wherever that happens to be. Here are some of the tertiary interactions that you should know about, the interactions between side chains. So one can be hydrogen bonds. Here's a exposed oxygen over here. Here's a hydrogen over here. They'll form a hydrogen bond. That's going to cause the polypeptide backbone to bend in a specific way. You can have ionic bonds, like shown over here at number 5. You can have covalent bonds. This is a very important bond. This is called a disulfide bridge between 2 sulfhydryl groups. And this is actually a covalent bond, and it really locks the structure in. And then you can have a bunch of hydrophobic amino acids that cluster together, sort of like a drop of oil. So the result is you get a complex 3-dimensional shape, like shown over here, as Milkshaker just said, a globular protein. Remember that one of the key themes of biology, which I haven't really talked about too much, is that form determines function. So the folding of a protein into a specific shape determines the function of that shape. And this is where like evolution and genetics feed back on one another. In other words, in the process of evolution, we have the production of proteins, the proteins that have a shape that enables them to better perform a function. Those are the ones that do their job best and get selected for. Holy cow, it's 7:42. How did that happen? Let's do nucleic acids. So the biological importance of nucleic acids, you need to know, of course, that DNA, the famous double helix, is the molecule of heredity. RNA is a single-stranded relative of RNA. It's the hereditary molecule in some viruses, but its key role is mostly about information transfer. It's about transferring information from DNA in a eukaryotic nucleus or in a bacterial chromosome out into the cytoplasm, where that RNA can be translated into protein. We just talked about how proteins get things done. Well, this is how DNA sends messages out to the cell to get those things done. RNA also plays a regulatory role. It can catalyze reactions in ribosomes and spliceosomes, microRNAs. And also, just to sort of have this filed away, ATP, which is life's key energy molecule, is an RNA monomer. So and that's probably not a coincidence. ATP is really essential to life. Okay, so powers most cellular work. You need to know about the structure of nucleotide monomers. Those include a 5-carbon sugar, a nitrogenous base, and a phosphate group. And the beautiful thing is that if you know that, you know the structure of ATP, because ATP is basically a 5-carbon sugar, a nitrogenous base, and then it's got 3 phosphate groups. DNA and RNA have different monomers that make them up. DNA has its sugar being deoxyribose. That's why it's deoxyribonucleic acid DNA. And DNA has 4 nitrogenous bases that can be A, T, C, or G. RNA has ribonucleotides as its monomer, and that's made of a sugar called ribose. Deoxyribose has 1 less oxygen than ribose. You see that there's an OH over here, and then there's an H over here, deoxyribose. And there are 4 bases, A, U, C, and G. So uracil substitutes for thymine. And that's important to recognize because you could get a question where, like, for example, the College Board will ask you to translate a sequence, and you need to immediately be able to determine if that sequence is RNA or DNA. Everything I just talked about. Okay, DNA structure. It's got 2 nucleotide strands. Within each strand, the nucleotides connect to one another by sugar-phosphate bonds. So what I'm talking about is sugar, phosphate, sugar, well, there's a phosphate that you couldn't say, sugar phosphate, sugar phosphate. And I actually have a rap called DNA Fantastic where I actually sing sugar phosphate, making up the sides of the ladder with nitrogenous bases on the inside, with the bases like the rungs, together with deoxyribose sugar and a phosphate, form a nucleotide. I think that's right. Yeah. The two strands connect with one another by hydrogen bonds between complementary nitrogenous bases. And you need to know those rules. A bonds with T, C bonds with G in DNA. Always getting ahead of myself. And the two strands are anti-parallel. So DNA can only form, like a base can only bond with another base if they're oriented relative, upside down relative to the other. So the same anti-parallel thing that I was just talking about with a relationship to beta-pleated sheets, that's what's going on right here, okay? This is just a little thing to tell you. If you've downloaded the checklist, like I told you to do, it's at apbiosuccess.com/checklist, then you'll note that like as you go through it, there are all these links, and those links take you to tutorials on learn-biology.com. And again, remember what you're doing over the next 7-8 days. Find the gaps, fill the gaps. And the most efficient way for you to do that is through the offerings that I have tonight, the checklist, and then the interactive stuff that is on learn-biology.com, which, you know, it's the website that guarantees your AP Bio success. And wow, here I am giving you a commercial, and it just appears for me. I didn't know that that was going to happen, but learn-biology.com, it's got tutorials, quizzes, interactive diagrams, games, comprehensive AP Bio reviews that we're going to go to right now. Right now, we're going to go to those comprehensive AP Bio reviews. And if you sign up for learn-biology.com for $24.95, you get the Biomania AP Bio App. And, you know, I'm not really looking at my email right now, but like I should be seeing like everybody here who isn't already signed up. You should be signing up for learn-biology.com, especially since now we're going to do really the fun part. We are going to go to learn-biology.com. That's the main page. And if you click AP Bio Review, you go to our AP Bio Review system. And let's just think about what we did. We just reviewed Unit 1 of AP Bio. So let's go AP Bio Multiple Choice Review. And we're going to start the quiz. And good. And here's what you could do. If you found that you had some gap in Unit 1, well, if it was a big gap, you could just do all of Unit 1. If it was a specific gap, you could just do part of it. And, you know, does anybody want to weigh in and like say, oh, I'd like to do proteins, I'd like to do nucleic acids? You know, I got a lot of questions. Carbohydrates, it's student choice. So you name the topic in Unit 1 that you want to review, and that's what we will do. So anybody have a preference? Proteins, nucleic acids, anything? We see some multiple choice questions, get some challenge, be a biomanian, make it happen. Proteins and nucleic acids. Sophie, thank you so much. Everybody else like Sophie's idea? You got 10 seconds to weigh in with something else. 9, nucleic acids, milkshaker. Love it. Love the interaction. Thank you so much. You are all indeed biomanians. I know that some of you are more biomanial than others because some of you are actually subscribing to learn-biology.com, which is a wonderful thing. I'm going to click proteins and nucleic acids, and we're going to click begin practice. Okay. This is the kind of question you get on learn-biology.com, and you can do this all day long if you purchase a subscription. So for $24.95, that's what you get. When DNA melts, its two strands separate to become single strands. I'm going to make this bigger. I'm going to go off screen. I think if I do that, does that do it? Yes. Okay. I just have to remember to come back. When DNA melts, its two strands separate to become single strands. A researcher is comparing the melting point of DNA from two different virus species. Both species store their genetic information in the form of double-stranded DNA. The DNA from species 1 melts at a lower temperature than the DNA from species 2. Which of the following best explains the difference? What a great question this is. Okay, so use the diagram below for reference, and that means carefully look at this diagram just for a minute. Just take it in. So species 1 had more adenine-thymine pairs than species 2. Species 1's DNA is longer than species 2's DNA. The two species' DNA was of equal length, but species 1's DNA had more sugar phosphate bonds. Species 1 uses deoxyribose in each nucleotide, while species 2 uses ribose. So the way that we do this, folks, if you're joining us for the first time, is that, first of all, I'm going to make this a little bit bigger. Go like that. And if you think the answer is the first one, put in a 1, put in a 2 for this one, 3, 4. So what's the answer, folks? Anybody else? I see an answer from Lily. Okay, now Sophie. Anybody else? We've had so many people here tonight. Aaron was here, Milkshaker was here, Vera was here, Ella was here. Anybody else want to answer? It's okay. Boy, and I am loving that name who just came in, but I don't know what language that is. I think it's Korean, but I don't really know. But anyhow, welcome. I'm so glad. I wish I knew how to say your name. And maybe you can give me something. Okay. So indeed, it is 1. Now, can somebody say Korean? Okay. Fantastic. Got that. Can anybody say why? Can somebody write just like a short sentence explaining why 1 is the answer? Why would more adenine-thymine pairs? Holy moly bio. Okay, holy moly bio. Fantastic. Why would more adenine-thymine pairs lead to a lower melting point? Everybody got that? I was scratching my chin. Nicely done, Lily. That's exactly right. So GC bonds are stronger than AT bonds because there are 3 bonds between G and C. 3 hydrogen bonds hold those 2 DNA monomers together, while only 2 hold adenine and thymine together. So that's why 1 was the answer. Fabulous. GC pairs have 3 hydrogen bonds connecting them. So again, this is what you get with learn-biology.com. You get a feedback. You get a pat on the back because let's say you guessed. Now you'd see the answer. Okay, let's do another one. Okay, good enough. The diagrams below represent various types of biological molecules or subunits of biological molecules. Which one is synthesized at a ribosome? A, B, C, or D? A, B, C, or D? Holy moly bio, good job. All right. Now, I'm seeing that the consensus is A, but I just want to show you something that's cool about Learn-Biology. Let's say you had a gap in this area and you clicked B. You get a hint, and look at the hint. B is a nucleotide. All right, so it's telling you that's a nucleotide, right? It's not synthesized at a ribosome. Here's a hint. Look at the diagram below. What is 1? And you can see if you look closely that 1 is a string of amino acids, but I don't tell you that directly. So this is how learn-biology.com builds understanding, right? So that's how you wind up getting it. So let's do another question. And also, just so that you know, if you were doing learn-biology.com at home, this question would go to the back of the stack. You'd get it again. So you wouldn't get out of that set of questions until you ultimately got it right. So it builds learning and it develops understanding. All right. This is really basic stuff that would not be on the AP Bio exam. Though the other two questions, certainly the first one is like an AP style question. I don't know about the second one. And this one isn't. This one is just like general knowledge question. And what do you think? Lily, thank you. Anybody else? Anybody else? I got Lily, I got Sophie. Okay, good job. All right, everybody's got that. Nucleic acid, way to go. Okay, in the diagram below, Y, letter Y represents, so the phosphate group, deoxyribose sugar, nitrogenous base, amino acid. Let's see it. Just do 1, 2, 3, or 4. Okay, I'm seeing it, and indeed it is. But let's try what Holy Moly Bio said, phosphate group. Okay, now here's where you get, again, so milkshaker, just so that, I'm sorry, Holy Moly Bio. The phosphate groups connect the deoxyribose sugars, and these sugars are represented as pentagons. So again, how does Learn-Biology work? It gives you a hint, and then it guides you toward the right answer. It builds understanding. Okay. In the 4 diagrams below, dashed lines represent hydrogen bonds between nitrogenous bases found in DNA. Which of the following statements is true about the diagrams? Of the 4 base pairings, GC and AT is possible. GT and GA cannot form the bonds shown. Second choice, GC and AT are the correct pairings. GT and GA are pairings that can lead to potential mutations during DNA replication. Third choice, all 4 base pairings are possible and regularly occur in normal DNA. GT and AT are more common in eukaryotes. GT and GA occurs regularly in archaea. And then the fourth one, none of the base pairings are possible because nitrogen does not participate in hydrogen bonding. So this is the real thing. This is the real thing. So what do you think? Okay. Anybody else? Okay. Why not? Let's go with Sophie's response. While the base pairing rules in DNA are G bonds with C and A bonds with T, the GT and GA base pairings are possible. They are possible. Knowing that, see if you can find a more accurate statement. And here's where actually I wish I had drawn this a little bit differently. AT and GC, they are the right width to fit within the helix, and they don't distort the helix. These other bonds would cause some distortion, and they could potentially mess up the process of DNA replication. So the answer indeed was the second one. It was that GC and AT are the correct pairings. GT and GA are pairings that can lead to potential mutations during DNA replication. Okay, folks, I have a question. In future meetings, more teaching or more quizzing? So how about we'll do a little poll right now. Would you prefer more teaching, like what I did for the first 40 minutes, or more quizzing? Teaching or quizzing? Let me see some results. Anybody, any ideas? More teaching? Yeah, I might do a show just about FRQs. Quizzing with FRQs. Quizzing with FRQs is going to be tough in this format. Okay. Let me think about how I can do FRQs, because I can do like College Board FRQs. They're all public. I don't think they're going to care. And I can do my FRQs. And then you maybe would type your answers into this, which is what I did like when I taught during the pandemic. A balance is good. Okay, so folks, here's what I want to do right now. We're on learn-biology.com, and here's what I'd like you to do. I would like you, if you're not currently subscribed to learn-biology.com, I'd like you to do this. I'd like you to go to learn-biology.com/AP Biology. It's like I'm doing. All right, I'm putting it in the chat. Okay, so there it is. And, when this ends, I'd like you to call over your parent, look at this. And I'd like you to look at the features of the website. I'd like you to watch this testimonial video by a student who used learn-biology.com and had some things to say about it. You can read these testimonials. Just read the thing, and then when you get down here, this shows you about all the features. Each one of these is kind of clickable, and you can see all the stuff that you get. So sit down with your parents and go over all the stuff that you get with learn-biology.com. And then I'd love it. It would mean a lot to me. But don't do it for me. Do it for you to sign up for learn-biology.com. Again, it's a $24.95 monthly subscription, but you're not going to pay me $249 over the course of the next year unless you get really hooked on it. You're going to pay me $24.95 because you can cancel your subscription anytime, and you're going to cancel at the end of the month. So I want you to go to, again, learn-biology.com/AP Biology. And I want you to learn about the website. I want you to sit down with your parents, because they're probably the ones who are going to pay for the site. Tell them that this is a website that can really support your learning. And then what I'd like you to do is, hit that, go to AP Bio Review, take them through the questions. Note that you can, I have, how many do I have? I have 139 practice FRQs for you. So show your parents what you get, what's available for you. I would really love it if within like the next 5 minutes after this show, I have 5 people signing up, because it's for you. It's going to get you to learn so much, and it's going to get you to that place where you want to get to. You're a biomanian, 4 or 5. I also want to let you know about one more thing. We're going to jump back to the slideshow. And I want to let you know that, where is that? Boy, I thought I was going to get a lot further. If you would like to join me tomorrow night, you can join me on Zoom. But the Zoom meetings are only available to Learn-Biology subscribers. So I really hope I see you there. These are there's a schedule of live reviews, and tomorrow night, just think about doing this, where we will be able to do FRQs really easily. We'll really be able to go into depth. So this was a pleasure. This was so fun. I am really honored that you chose to spend this hour with me. Thank you so much for joining me. Sit down with your parents and take them to learn-biology.com/apbiology and tell them that this is the support that you need to get the score that you're creating, to get the success that you want to achieve. And I can be with you in these YouTube sessions, but I can give you even more support if you sign up for learn-biology.com. So I hope everybody does that. It's been such a pleasure being with you tonight. I'll see you at the next live review. And I hope I see you tomorrow night on Zoom. To do that, go to learn-biology.com/apbiology. Sign up for your subscription. Good night, everybody. Be well. Thank you.